Okay, hi everyone. This is our class meeting for Thursday, June 4th. We're not able to do it live, and what I was going to do was to review for your lab quiz slash exam. And just so you know, I think I kind of wrote in your email that, it'll be like 10 to 20 questions, though this set of material is probably a little more lengthy, so it may be more like 20 to 30 questions. They're all multiple choice. They're going to come straight from the labs and what you did, very straightforward. Okay, so as long as you review the lab assignments, you'll be fine. Okay, so let me just sort of quickly review, point out where everything is. As of now, I did grade everyone's metric lab assignment and also the metric and microscope assignment. So both of those have been graded. Also, you have due on June 4th, right? The chemical composition of cells, and then the two separate parts for the cell structure and function lab. So I'll review them as well. So again, I'm not going to post answers. For the most part, again, I pointed out if you were missing or something was wrong when I graded them, but for the most part, everyone did pretty well. A lot of times when points come off, it's because things are missing. If you then complete them and email me, then I'll change your grade. Okay, so let me just do a quick review. And then I'll be on my way. As always, if there are questions, please post them in the question and answer forum. Okay, so let me share my screen with you. I'll pull up just, a couple of the lab documents essentially. Actually, for the metric and the microscope lab, I am going to probably use somebody's completed lab just to kind of quickly go over it. The one thing I also just want to show you real quick is that there is a I guess I'll show you where that is on Moodle, just so that you know. There is a review sheet for the lab exam, okay? So again, just because I always kind of update you guys as we have class meetings, we are in, sorry, let me just check my dates and stuff like that, right? So for this week, we are in modules 7 through 9, right? 7 through 9 is going to be completed. That's the other thing. I will also have the recorded lectures uploaded for both chapter 7 and chapter 8, okay? But we are we're in modules 7 through 9 for this week. And so that will be that enzyme lab and chapter 7 and 8. I will put up a recording for that enzyme lab as well, okay? So those are upcoming. Everything that I'm going to review was from the previous modules, right? Again, remember your labs for the chemical composition and the cell structure and function labs are due on June 4th, okay? And that's all part of lab exam 1. So I just wanted to show you, again, I have lab exam / quiz 1. Like I said, this one will probably be about 30 questions or so, multiple choice, 20 to 30 questions. And it will be available starting on Sunday, okay? There is a review sheet here, okay? So you guys can take a look at that review sheet. Pretty much, I probably won't be going back and forth to that review sheet because I'm just gonna go through the labs and just sort of hit some of the sort of high points of the labs. But remember that review sheet is there, okay? So again, in terms of what's on the lab exam / quiz is all on this review sheet. It all comes from your lab assignments, okay? Remember, you don't need to complete this as an assignment. This isn't one of your assignments. It's just there for your reference. All right, so again, like I said, I'm not going to keep referring back to that. So let me just go back and start with the first assignment, right, which was the metric assignment, right? So basically just doing conversions. So there will for sure be a couple of conversions, just straightforward ones like this on the lab quiz. I mean, I said all multiple choice, but I guess some of them could be worded so that it's not just simply plug in the numbers, right? Because you can easily, obviously, just Google, what's 2.3 meters in centimeters, and you can just get the answer in 2 seconds. So I may word them differently to try to minimize that. But bottom line is basic conversions, right? And so just to kind of review, this was your first assignment, right? Just practice. The first sheet talks about, deciding whether, you're going to move your decimal place to the left or to the right, right? Depending on whether you're starting with a larger unit or a smaller unit. Okay, so some examples here. If we're gonna convert 2.3 meters into centimeters, right? It's 230 centimeters. So remember there are, oh, wait a second here. Yeah, right? So there are 100 centimeters in a meter, right? So we need to move our decimal place 2 places to the right. So 2.3, right? 1 decimal place would be 23, and 1 more is 230 centimeters. We should also be able to do those conversions, you know, going from centimeters to millimeters, from millimeters to micrometers, just like we had the examples here. Also then doing similar types of conversions with units of volume, right? So 1.8 liters is equal to 1,800 milliliters. Why? There are 1,000 milliliters in 1 liter, okay? So it's 3 decimal places to the right, okay? So again, remember when you're going the other way, so if you're starting with a smaller unit and then moving to a larger one, your decimal place is going to move to the left, okay? So again, there are 1,000 milliliters in a liter, okay? So the liter is the larger unit. So again, if we're going to convert 53 milliliters into liters, we must move our decimal place this way to the left. Okay, so just a little bit of review, pretty straightforward. There will definitely be a couple questions with conversions. Okay, so the next assignment was the metric and microscope assignment. So again, as long as you're reviewing the lab, you're fine, okay? So just a couple things. Again, there was more conversions here, right? So we want to be familiar with the conversions. This was all basically converting. So I'm not going to really go through that again. It was just a matter of kind of practicing your conversions again, doing some measurements, right? When we got to the volume, I talked a little bit about different types of glassware that you would use to measure volume. We talked about a graduated cylinder and a beaker. We should be able to like recognize those, right? So if I showed you a picture of a beaker, you should be able to tell me it's a beaker, not a graduated cylinder. We talked about a transfer pipette, okay? So those are things just in terms of identifying. For this question, number 3, a couple people missed this, and I think this usually happens pretty much sort of every year. It's just kind of a simple question. Sometimes we sort of misinterpret it. So if you want to know, you know, what the total volume of that test tube is, it's simple, right? Just fill the test tube all the way up with water, pour it into a graduated cylinder, and then look to see what the volume is, okay? It's not really meant to be more complicated than that. All right. So again, for conversions, we should be able to convert from Fahrenheit to Celsius. You know, it's not like you have to memorize the equation or anything like that. You'll have it, you'll have access to it. So just kind of a heads up to that. In terms of the microscope, right? There's a couple definitions and stuff like that in the introduction. Let's make sure we know those definitions. For instance, it asks about 2 types of microscopes that view the surface, right? Everyone pretty much got this right. The scanning electron microscope, which is obviously going to be a lot higher magnification, right? Or in higher resolution than the, oh, so this is actually wrong. Sorry. The surface is scanning electron microscope and the dissecting scope. So the dissecting microscope, you can only see the surface. It's at a much lower magnification than the scanning electron microscope. Okay, so just to point that out, because they're actually, I think to be honest, I think most people got this right, but here's an example of sort of probably just not being too careful about the question. So make sure you read through this and understand that. So scanning electron microscope and a dissecting scope will allow you to view the surface of an object, okay? If you're talking about sliced objects, right, and being able to kind of get a little bit of sort of going through the depth, that's going to be the compound light microscope, right? Remember I showed you slides. That's the microscope that we would use in lab. And then it would be the transmission electron microscope. Okay, so these are just mixed up a little bit. So again, this is why I'm going over it. I just wanted to kind of point out, make sure everything's clear to everyone. Okay, so again, yes, if we're going to talk about high resolution, it's the electron microscope. All right, we should know how to identify the parts of the microscope, the compound light microscope, right? And so I will definitely have a diagram or two of the microscope and ask you to identify the part. Also, I can ask you about function, right? Like what's the function of the coarse adjustment knob here, or the condenser, okay? And so these are all again listed in that lab. I mean, in terms of the inversion and that kind of stuff, those are really just exercises to understand the operation of the microscope. I may ask you about the definition of parfocal. Total magnification, I could give you an example saying you're using the low power objective, right? That has a magnification of 10x. Your ocular lens has a magnification of 10x. What is the total magnification? Okay, so we should know that that is 10 times 10 equals 100. Okay, so again, this table should be filled out so that we understand that total magnification. If you were using scanning, right, the objective lens would be 4x, say. I would tell you what the magnification is. So say the objective lens is 4, the ocular lens is 10, it's always 10. 10 times 4 would equal the total magnification of 40x, okay? So again, make sure you know total magnification. That is also going to be on the lab quiz, okay? This was just an illustration of putting in the, what one was on top, blue, yellow, or red. Again, that's sort of meant to be a demonstration so that you understand the sort of depth that you can kind of figure out while operating the microscope. All right, we should be able to identify or label a picture like this in terms of the, this is not a cell wall, right? This is a plasma membrane or cell membrane, cell wall, right? Yeah, I guess it is cell wall. Sorry, I thought for a second it was the cheek cells. It's a cell wall, right? That's what you're actually able to see, and it's actually a lot more clear and prominent than the cell membrane. And then also the nucleus, right? Because these are onion cells, these are plant cells, right? So if I ask you, if I showed you this picture, I could ask, is this plant or animal, right? So those are things that we should be able to distinguish. So again, here now we have the animal cells, right? Plasma. You got your nucleus here, right? Showing you the cytoplasm, and then the plasma membrane on the outside, okay? All right, and so that's pretty much it for that microscope lab. Let me now, sorry, what happened here? I got another, oh, this is so annoying. One second. Let me now move over to that next lab, which is the chemical composition of cells, okay? So in terms of this lab, what I'm going to kind of ask you on is basically about the tests, right? I could ask you also about negative and positive controls. Remember, I talked about that. But mostly it will be, what does the Burette test for? It's proteins, right? If I showed you one of these pictures, right, and I basically, showed you this that was blue, and make sure it's clear. Well, it starts off blue, right, which it tells you here. And then I showed you another test tube, right, after the experiment that was still blue, you would say that was a negative test, right? If it was purple or pink, then we know it was positive for proteins, okay? So those are the types of questions I'm gonna ask, and I probably will use pictures to say, is this positive or negative, right? What does that indicate to you? So remember, for testing for carbohydrates, we had 2 different tests, right? So test for starch, iodine, turns blue-black, okay? Again, we should understand that. And then we did a test for sugars, the Benedict's test, okay? And again, same thing. If I show you a test tube, right, that's green, yellow, or red, we know that's a positive test for sugars, okay? All right, and that's pretty much it for chemical composition. All right, so now let me go for cell structure and function. For the first part, pretty straightforward, diagrams of the cell, right? Identify the parts of the cell, major parts of the cell, know the function of those organelles. You need to know that for your lecture exam that you're taking now anyway, okay? So you should already know that. So in terms of identifying parts, right? Remember, you're just basically writing in the function here and then writing in these parts. and also these, and adding them into this diagram here, okay? So I will either use this diagram or a similar diagram and ask you basically, you know, is this the ribosome or is this the mitochondria or something like that, right? So we should be able to identify parts. And then same thing for the plant cell, all right? And same thing, if I showed you these this diagram and asked you, is this a plant cell or is this an animal cell? We should know that, okay? So that's pretty straightforward for that. And then the very last lab that's part of this lab quiz / exam, right, is the diffusion and osmosis lab. And remember, part of that was a, you know, a video demo. So definitions, yeah, I can ask you about definitions that are here. That's all part of that background information, okay? I will definitely include some pictures like this, right? And basically kind of ask you what I'm asking you here, okay? Also knowing that these are plant cells, right? And these are examples of animal cells, of red blood cells. And so, right, what if I showed you this cell here, I might ask you what type of solution is this cell in based on its appearance, right? And so here we know that water's being drawn into that cell, right? So this is in a hypotonic solution. There's more water on the outside, gets drawn into the cell and causes that cell to swell, okay? If we look at these plant cells here, we can see that plasmolysis is happening here, right? Everything's getting pulled to the inside or to the center of the cell because water's being drawn out and it's shrinking, right? In terms of, you know, the hypothesis and stuff like that, I don't know that I'm going to ask you about the hypothesis or anything like that, but I guess the whole point of this is to understand or make the connection between the size of a cell or the surface area of the cell and how much diffusion can happen. This is more cell size, right? If the size of the cell is too big, right, materials won't be able to diffuse throughout that whole cell, okay? So it needs to be small, have lots of surface area. And that was the whole point of this experiment here. In terms of the diffusion across the plasma membrane, I think we really need to understand the basics of that experiment, right? And I prompt you by asking you some things here. The Benedict's test, we should know what the purpose is, right? To test for sugars. And wait, let me just check my notes here because I did have notes because this is from the video. I just want to just kind of go over this just quickly with you guys because I think it's important, even though I know some people still haven't handed in the, shoot, where's my notes? One second, I have them. I know I have them right here. Yeah, okay. Yeah, so for that dialysis experiment, right, in that demo video, basically you had a couple different beakers set up, right, with that dialysis bag in the middle, and that dialysis bag is your model cell, right? And the dialysis bag serves as a semi-permeable membrane, okay? So that means that some molecules are going to be able to cross through that and some aren't, right? And so when we're thinking about this, we want to just purely think about based on size, okay? Now, when we talk about the regular plasma membrane, you have to take into account things like whether the molecule is hydrophilic, right, or hydrophobic. But for this purposes and illustrations of this experiment, it's about size, right? Larger molecules maybe cannot cross, but smaller ones might cross, okay? And so the way that experiment is set up is to try to figure out, you know, which ones were able to cross and which ones were not, okay? So I believe, you know, there were sort of 4 different situations that you had in that video. You had a beaker, right, filled with water. In one, you had iodine in the outside in the beaker. And then you also had, let me see if I can draw this. One second, I'm going to stop share and see. I mean, I'm not very good at drawing and I'm probably really bad at drawing on here, but let's see if I can just kind of, it might be just easier to do it that way and then we'll be finished. So bear with me because it'll probably be a terrible drawing. Like I said, even with like a regular board, I can't draw. But so here's your beaker, right? And it's filled with water. So we can write water here, I guess. Maybe I'm not too bad. It's not going to be this. I'm not going to make a subscript or whatever. So here's your water. And then say this one also had iodine. Okay. Then what happened to my drawing tool? Draw, right? Then we had the, hey. Okay, then we had that dialysis bag that was in here, right? And inside that dialysis bag, so say for example, in this one, you had starch, okay, it was inside. Okay, so starch inside the bag, here's your dialysis bag. On the outside, you had water and you had iodine, okay? So I'll just kind of go through this just with this first one, and then you can think about all the 3 different other situations that you had in that demo and think about what might be happening. So the idea is if you had starch, okay, you have starch inside. Starch, remember, is a complex carbohydrate. It's a really big molecule, right? And you have iodine on the outside. And so this then makes this sort of an amber sort of color, okay? Remember that iodine is a test for starch, okay? So if the starch is able to cross through this semi-permeable membrane and end up in the beaker, what would happen to the color of the water on the outside? It would turn blue-black, right? So that's how you would determine if that starch was able to move out. Okay. Now, again, I told you it's a really large molecule, so you might think, probably it's not able to. Okay. Now, they also then had you sort of measure also the weight of the bag, right? Because then you can see if water went into that bag, right? More water ended up in there after than before, right? Because we can think about osmosis, right? And the movement of water, all right? But if you want to kind of just like think about the movement of the molecules, you had some tests, some chemical tests that we did in the previous lab that can help you determine that, okay? So if starch got out, this would turn blue-black. If this doesn't turn blue-black, it indicates to you that starch did not, was not able to cross through, okay? So that's the sort of idea there. Though there was also another one where, right, because just to point this one last one out, right? There was another one. Here's your dialysis bag. That's so terrible. And in this one, right, you had obviously water, okay? And then inside here, you had, dextrose, right? So a sugar, okay? And one of the tests, right, so there's dextrose inside, there was water on the outside. And then at the end of the experiment, you did a test, a Benedict's test of this water, right? So again, if you had a positive Benedict's test, it would indicate that this dextrose left or was able to cross that semi-permeable membrane, okay? So anyway, I just wanted to kind of make sure I sort of provided a little bit of more explanation with that experiment. Like I said, the first part of it is really just about the relationship between the size of a cell and how much diffusion can occur. I think this one with the dialysis bag is a little more complex, but I also think it's a little bit more, I mean, I like the experiment because I think it really illustrates this sort of semi-permeable nature of a plasma membrane, okay? And then also brings in some of those chemical tests that I did in that previous lab. All right, so again, if we're going to talk about sort of movement of things across a membrane, you can talk about the movement of water across the membrane, which is osmosis, right? And we talked about what happens when different types of cells, right, like plant cells or red blood cells, animal cells, are placed in different environments, right? How much water moves in, or is the net movement of water into the cell or out of the cell, and how does that affect the health of the cell? Here, again, is sort of an illustration of that using obviously a sort of man-made setup, okay? And if you want to, what I kind of focused on was following the movement of the solutes, right? But as I said, you know, they also at the end of the lab weigh the bag, okay? So if this dialysis bag at the end of the experiment weighs more, then it indicates water from the beaker traveled in to the bag, okay? If it weighs the same or it weighs less, right? If it weighs less, then it would indicate that water was moving out. Okay, so you're both looking at, from that experiment, you're looking at the movement of solutes, but you're also looking at the movement of water. All right, and so that's, again, illustrating what's happening in the cell. Okay, so let me get rid of this because it's pretty sorry looking. And that's pretty much it, guys. So that's a review of, you know, a decent amount of lab material. Unfortunately, we didn't get to do it ourselves, but I think you guys hopefully got the major concepts from it. So that lab, like I said, exam/quiz will be about 20 to 30 multiple choice based on all of those labs and those lab assignments that I just went over. Thanks, guys.